Welcome to Ask the Expert. This is a brief and informative and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event and we're going to post it on the Sugar Science YouTube channel shortly after the presentation. Feel free to engage with our guest with questions in the chat or raise your hand. And coming to us today from Boston University Medical School is Dr. Barbara E. Corkey. She is professor emeritus there, and her current focus is on developing clinical basic behavioral collaborative multi-PI projects to explore novel approaches to understand and treat metabolic diseases. Dr. Corkey was the Zoltan Cohn Professor of Medicine and Vice Chair for Research in the Department of Medicine at Boston University. And she's been a leader in the fields of diabetes and obesity research for over 50 years with about 200 related publications. Wow. And 40 years of continuous government research support. Finally, she has received numerous honors, including the NIH Merit Award, the National Honorary Membership in Iota Sigma Pi, National Honor Society of Women in Chemistry, Women in Science Lecturer at Boston Museum of Science, the George Bray Founders Award of the Obesity Society, and the Charles H. Best Lectureship and Award. University of Toronto, and the Banting Medal for Scientific Achievement from the ADA. The last is really huge. Welcome, and thank you so much for joining us today. I'm really excited to hear this talk.
Well, wonderful, and thank you very, very much for inviting me. As you know, type 1 diabetes is not my field. I've come to it through some very odd circumstances. And what I know about type 1 diabetes is that it's an immune disease.
I'm sorry, I think there's some kind of shuffling going on. It's a little bit, yeah, that noise.
Yes. Oh, okay.
Maybe the microphone is just too close, but sure. We can edit that out. That's better.
Okay, so what I know about type 1 diabetes is that autoantibodies are early indicated that type 1 diabetes may progress, and that beta cells are destroyed by an autoimmune process, and that so far immune therapy has not been very effective. And I think, interestingly, the cause of autoimmunity is not known.
Yeah.
And we also know there are environmental factors that enhance the likelihood of getting type 1 diabetes, and those influences are not known. So I'm going to present a hypothesis that's based on my work that was never intended to address type 1 diabetes, but it comes together in a rather interesting way. And it's a model of what might cause autoimmunity. And the good thing about this model is it's very testable. And if it proves valid, and that definitely needs some studies to prove it valid, there are some obvious potentially effective therapies that could prevent further development of type 1 diabetes. So the idea that I'm presenting today came to me based on work in 3 totally unrelated areas, unrelated to each other and unrelated to type 1 diabetes, as a matter of fact. The first is sepsis, the second is wound healing, and the third is basal hyperinsulinemia, that is the precursor to type 2 diabetes. And so the studies in sepsis, whoops. All right, can I make it go? Yes. The studies in sepsis were done a very long time ago. And now, just to refresh everyone's memory, sepsis is a systemic inflammatory disease that includes inflammation, fever, anorexia, a drop in blood glucose, increases in cytokines, including TNF-alpha, a drop in the energy state, the ATP-ADP ratio, and often vomiting, low insulin, increased fat, and ketones. And so what we learned from these studies that's going to be relevant to the development of this model is that TNF-alpha, which rises in these systemic inflammatory diseases, inhibits fatty acid oxidation. Now, this is bad because under these conditions where food is not being taken in, fatty acid is the only energy source. And under extreme circumstances, inhibiting the only energy source will ultimately lead to cell death. So the take home from this is that TNF inhibits fatty acid oxidation. So we're going to remember this through the next two areas. The next study was done by a student, an MD PhD student of mine, who was interested in surgery and was curious to know why wound healing was defective in diabetes. And so in order to study this, we obtained 20 cultures of human fibroblasts. 10 of them were for control subjects, and 10 of them were from type 1 diabetics. And these were pair matched, same passage, et cetera, and treated over many passages through this with the same media. So they had not been in a patient for a very long time, and they were not being exposed to anything unusual. They were all being exposed to exactly the same thing, is what I intended to say. And the first thing that we found was when we stained these cells that were grown in the presence of fatty acids, the control cells accumulated some lipid, the top A and B. But if you look at that, the lipid was mostly in the plasma membrane around the outside of the cell. However, the diabetic cells were different. Under basal conditions, they had more lipid in them, but under growth in fatty acids, they became full of lipid. And this was totally unexpected. We don't know what that meant. The student was interested in wound healing, and among the various factors that are involved, bradykinin is a good agonist. It works, it interacts with a membrane receptor, and it elevates cytosolic free calcium. So the next big surprise that we found was that if we compared the control fibroblasts and the diabetic fibroblasts, there was no difference in their response to bradykinin. But if we treated them overnight with TNF alpha or IL-1 beta, a huge difference evolved. And you can see, I'm showing here the individual patients. And so on the left are the controls, on the right are the diabetics, and there's almost no overlap. There were a few of the controls that had relatively higher responses. And in evaluating those, we found that they had first-degree relatives with type 1 diabetes. So this is an amazing, huge, in studying human samples, they're never very similar. They usually cover a huge range of values. So this much separation was a big surprise to us.
Yeah, it's remarkable.
And it's not necessarily bad, it's different.
Mm-hmm.
But now here is a sort of summary of the findings that we got with these diabetic fibroblasts. Something similar happened in the controls, but nowhere near as markedly. And so what you can see on the, can I make my thing go? Yeah. On this lowest trace is this is measuring cytosolic free calcium in real time. And is a transient rise in calcium in response to bradykinin. Now, if the cells have been treated overnight with TNF or IL-1, that response is much bigger, and that's what I just showed you. But if they were treated overnight with fatty acid, it was even bigger still. And when the two were combined, it was humongous, almost 10 times higher. And this difference was very consistent across all of the samples that we had, to our shock and surprise. Whoops, I'm going ahead of myself. Let me just summarize then what I want the take home from these studies in wound healing is that type 1 cells from type 1 diabetics are more sensitive to TNF and free fatty acids than control. And what that means, we don't know. It could be, you could imagine it might be beneficial because in wound healing, that might make them heal better when under some circumstances, but we didn't know what it meant. It was just very curious. Okay, now on to the third area, and that is the beta cell studies. And beta cells are, or subjects who are going to develop type 2 diabetes are hyperinsulinemic at basal glucose levels. That insulin level without stimulatory glucose is sometimes as high as 5 to 10 times greater than a lean normal control. So they, and we know a lot about how glucose stimulates insulin secretion, but there's been very little done on what stimulates basal insulin secretion. But what we do know is that it's not the same thing as what stimulates glucose stimulated. Calcium is not involved. And so we sought to find out what might be involved. And this is 2 slides that I'm going to show you that illustrate this. So we have here on the left, normal glucose, 3 millimolar, and then we increase it to 20 and you get a big increase in insulin secretion. Now, if we keep at 3 millimolar and we add a little peroxide in, we see that there's a concentration dependent increase. The only variable here is ROS, or peroxide, which is the predominant form of reactive oxygen species in cells. And we can do the same thing. We've done it over on the right panel using DEM, which generates peroxide inside the cell. And again, at 3 millimolar glucose, increasing peroxide generation increases insulin secretion. So this is one thing that we could think about that could impact. The other thing that is going to help us tie all this together is lipid. And I showed in the first slide with the fibroblasts that fibroblasts from type 1 diabetics accumulate more lipid than others when exposed to fatty acid. So these now are beta cells exposed to excess nutrient, grown in excess nutrient, and then tested at normal glucose levels. And here we have insulin secretion at 4 millimolar glucose in these cells. And these cells have been grown for 24 to 48 hours, either in 5 millimolar glucose, 11 millimolar glucose, low glucose plus fatty acid, and high glucose plus fatty acid. And on the bottom here, I've measured the lipid content of these beta cells. So in response to the excess nutrient, they accumulate lipid, just the way those fibroblasts did. And that it correlates very nicely with the lipid content and the basal insulin secretion. So we have 2 things here now that relate back to what I started telling you about, that peroxide or ROS, or which is generated through inflammation and cytokines, causes insulin secretion, and lipid accumulation causes insulin secretion. So with those 2 things varying only, and none of the other parameters that we know that are involved with glucose stimulated insulin secretion, we could explain basal insulin secretion. And we also know that if we scavenge ROS with a ROS scavenger like N-acetylcysteine, we inhibit insulin secretion. So it appears that ROS is both essential and sufficient to stimulate insulin secretion without anything else. It's not as good a stimulus in a physiological sense as glucose is, but it could explain basal hypersecretion, at least in part. So now we come to the hypothesis. Beta cells respond to cytokines and lipids with increased ROS production. That's well known. Since inflammatory diseases often occur in individuals before the onset of type 1 diabetes, in fact, a lot of viruses, I think, have been studied as potential causes, but none of them have really panned out as the important cause. But with each one of these inflammatory events, there would be increases in cytokines and free fatty acids and consequent ROS production. So this would then lead to, let me see if I can do this, increased vesicle cycling. So this is, these granules come to the plasma membrane, secrete their insulin, and go back in, go round and around and around. And the more basal activity you have, the more this process occurs. So there'd be increased, so this would then increase this process with TNF and free fatty acids, the two conditions that we found. If TNF also inhibited fatty acid oxidation, it would also deprive of energy. So in the end, we would have increased vesicle trafficking, increased basal secretion, at least transiently during these events, and increased granule turnover, much as occurs in type 2 diabetes. However, in the absence of adequate energy, if we had the same process, TNF inhibiting oxidation, there would be defective granular protein processing. And this could lead to odd products that come from the processing pathway being secreted. It could also lead to presentation of proteins that don't normally have that much presentation to the immune system. And also the ROS production, the peroxide, would ultimately lead to cell damage. And so the hypothesis that I'm presenting to you, and that I'm encouraging people who might be interested to test, is that the cause of autoimmunity in type 1 diabetes is prior infection, or possibly also ROS-generating environmental toxins, that this would lead to increased vesicle trafficking, presentation of antigens to the immune system, insulin processing defects, and so you would expect more pro-insulin or partial insulin processed relative to C-peptide, which in fact does occur, and increased oxidative damage due to sustained ROS elevation. If this hypothesis is correct, it has some potential remedies that we could recommend. And first of all, the simplest thing would be to test the effect of increasing fat oxidizing capacity, because this very high lipid component would be diminished if the beta of all of the cells in the body, never mind the beta cells, were more able or willing or programmed to oxidize more fat. And in fact, children often have a very relatively low fat diet, a relatively high carb diet. So lowering the carbs, upping the fat, or in addition to that, there are drugs, fibrates like fenfibrate and bezafibrate, that induce fat oxidizing capacity. So it'd be interesting to see whether or not there could be a benefit to those. Of course, this benefit would want to be applied prior to the development of type 2 diabetes, not after the disease is in place. The other implication of this hypothesis, should it be proven correct, is that it would encourage people to treat infection in susceptible individuals very rigorously, and in particular to maintain blood glucose and energy supply. And then finally, I think another possibility is that inhibiting TNF binding to its receptor, prior to severe onset of disease might be useful. And finally, scavenging ROS, if the elevation in ROS, and this can readily be measured in the blood, is sustained beyond the period of infection. So there you have it. That's my model.
This is really interesting. I have a couple questions, and I'm sure that members of the audience do as well. I'd like to invite the audience first to ask a few questions.
Yeah, I could I say something?
Yes, please.
Yeah, it could be that the beta cells do not relax, and we and others have published that it's important for beta cells not to be too active, and maybe the relaxation is the most important thing, like in psychology, so to speak. And fatty acids making higher fasting or basal blood glucose levels would by itself be dangerous for the beta cells. So it could be a very good idea just to lower the free fatty acids. And the model could well be used, but it could also just be that the beta cells are not relaxing enough. And then the beta cells express more antigen, which is dangerous to do too much for the immune system and so on, and then the process could start.
Totally agree with you. I think it's easily doable and very testable. And I hope I'm not doing active research anymore. As you know, I'm an emeritus professor now, so I've stopped fighting with my trainees for grant money.
Yeah.
And
I'm still doing.
I've written a review on this that I'm about to submit to diabetes. And Carsten, I think I might recommend you as one of the reviewers, and I'm overjoyed to be criticized on this because it's a new area for me. But yes, I agree with what you're saying. And I think the other thing I didn't really talk about is that, and I know nothing about immunology, is that the different immune cells that are involved in the damage to beta cells are sensitive to lipids and cytokines. And I'm not sure how that would play in, but I know that there are people in the field who can figure that out, and it's testable.
As you can see, and active beta cells are more likely, it produce 1 million insulin molecules per minute or something very fantastic. And such a beta cells are more likely to come out with a wrong molecule which attract attention for the immune system. And this could, at the very end, start the process.
Yeah. So it's interesting in both type 1 and type 2, the proinsulin to C-peptide ratio rises. And in type 1 diabetics, even long-term diabetics, they have measurable proinsulin sometimes.
Yes, that's right.
And so this to me is amazing. What it says, it directs our attention to something that happens in the processing. And I think that I didn't talk about calcium, but calcium is screwed up also by cytokines. And so it depletes the ER of calcium, which is necessary for proinsulin or insulin processing, and also for many of the other events that occur in putting a granule together. So yeah, that's, I couldn't agree more. I think I think the model fits, as far as I could tell, with available data. It isn't a proven model, and I don't want to overblow this. I wouldn't want to give a wrong impression or mislead anybody.
And what we do with the phenofibrate, that is to giving to newly type 1 diabetic patients, or even better, maybe to antibody positive patients, which we haven't done as yet, to give phenofibrate to them. And it increases the amount of sulfatide, which seems to be very important for the beta cells. and actually it increases especially the long chain sulfatide, the C24 length of sulfatide, which is immune one, which stop the immune system, whereas the shorter one, C16 sulfatide, is doing a lot of physiological things in manufacturing insulin. It's a chaperone to insulin, but the C24 is the one to stop the immune system, and which has been published. You can just look up my name on sulfatide and so on. So, but what was the drug you, I didn't catch the drug you would like to give? It was some fibrate. What was it, Barbara?
Bezafibrate, which I think is available in Europe, but not in the United States.
Beta fibrate.
Yeah.
Yeah. Oh, I don't
It's
know.
actually got some beautiful data that is largely ignored, but in type 2 diabetics.
Yeah.
But I mean, the argument, the damage that can happen to a beta cell not handling fat adequately applies both to type 1 and to type 2.
Yes. Maybe you should see there's a parallel to, serious burns, when a kid or someone have a terrible burn, then there sometimes can come an episode of hyperglycemia, because insulin production should be very high, but too low for the demanding. And
right. It doesn't address many other, I mean, there are many other aspects that are fairly well established, but and I think my analogy was that what we found in fibroblasts, which have nothing to do with type 1 diabetes, really, it was just a model system, probably is true of all cells. Mm-hmm. It's probably, the dose response could be a little different, the sensitivity could be a little different, but importantly, I didn't mention this, the ROS scavenging system in beta cells is much less. And this is both good and bad, because ROS is a very important signal for fuel excess. And it's probably why the beta cell in a healthy individual responds very rapidly to an increase in fuel availability. But in excess, it does all these other bad things. So, I think most things in nature are kind of bell-shaped. There's the right amount, and then there's too much. And you need to be in the right part of the curve.
Yes. It's often a balance between, sure.
So I think a final word would be that the problem here in type 1 is autoimmunity. We know an awful lot about what happens, what the destruction is, and what goes on once the disease gets underway. Yeah. I think we need to have focus on the earlier times. What causes autoimmunity?
The prodrome.
And maybe my model is wrong, but I think we should welcome other models that are consistent with available data, because if we all who care about this focus on figuring it out, we're much more likely to find a solution than if we don't do anything.
I agree. And I think that the time is now. There's a lot of new tools in the space. There's a lot of new approaches. There's a lot more data. And so if we can understand the etiology of this heterogeneous autoimmune disease, it may open the doors to understanding many other types of disease like this. So, all right, then. I'll let you get back to your days. Thank you so much for joining. Really enjoyed
it was highly interesting.
It was great. I'll hope to see you again in the future, both of you.